So, I am going to explain this slide one more time. So, stomata are the tiny pores which are present in the leaves through which the exchange of gases takes place. So, each stoma has a pair of guard cells which controls the opening and closing of the pores. When water enters inside the guard cell from the subsidiary cells, so at that time that guard cells get swell up and turgor pressure get developed inside the guard cells, and because of that swelling, pores will get open up. Instead of that, when the guard cell lose water, it will get shrink and all the pores will get ultimately closed. So, both the diagrams in wide view I am going to show you. So, this type of structure we have already observed in the biology lab when we prepared the temporary mount of stomata, right. So, in the same way we have observed the diagram inside under the microscope. So, this is a wide view of second diagram. When the water get diffuses into the guard cell from subsidiary cell, which causes them to open because of turgor pressure. In hot and dry days, the guard cells have less water content because evaporation rate is more, and because of that they relax and stomata will get closed. Okay. So, this is the diagram showing the opening and closing of stomata. Then this activity is given in your textbook. The activity is activity to show that chlorophyll is necessary for the process of photosynthesis. So, first of all, take a potted plant having variegated leaves. So, it can be a crotted plant, it can be a croton plant. So, keep it in the dark room for 3 days so that all the starch is get utilized. Then keep it in the sunlight at least for 6 hours. Then take a leaf from plant and mark the green areas of the leaf on a sheet of the paper. Then dip the leaf in the boiling water to make it soft. Then dip the leaf into the alcohol and heat it in a water bath to decolorize it and remove the chlorophyll. So, everything we are doing to remove the chlorophyll only. Then wash the leaf in water and dip it in the dilute iodine solution. So, it will be seen that only the green part of the leaf turns into the blue black. This show that the chlorophyll is necessary for the photosynthesis process. Then second activity is the activity to show that the carbon dioxide is necessary for the process of photosynthesis. So, first of all, take 2 potted plant of same size and keep them in a dark room for the 3 days so that all starch is get utilized. Then keep the plant to separate on separate glass plates. Keep the watch glass containing some potassium hydroxide near to one plant to absorb carbon dioxide. Cover both the plants with bale jars and seal the bottom of the jar with Vaseline to make it airtight. Keep the plant in sunlight for 3 hours. Then take a leaf from each plant and test for the starch. The leaf of the plant kept in a jar containing potassium hydroxide does not show the presence of starch. So, this shows that carbon dioxide is necessary for the process of photosynthesis. Okay. So, because that potassium hydroxide will completely absorb the carbon dioxide content present in that jar. So, if carbon dioxide is not present in that jar, the process of photosynthesis will be impossible. So, that is the reason it shows that carbon dioxide is necessary for the process of photosynthesis. Then nutrition in animal, I have explained this concept, that is nutrition in amoeba. So, in the diagrammatic way I am going to represent it one more time. So, amoeba is the unicellular living being, and it takes food by forming finger like projection, that which is called as a pseudopodia. So, and forms a food vacuoles inside the body. So, inside that food vacuoles, food will get digest with the help of certain enzymes, and these digested complex food material which get converted into the simple form, it will get absorbed by the body. So there are 4 different steps in nutrition in amoeba. First is a ingestion, then digestion, then assimilation, and then egestion. So ingestion is taking the food inside with the help of pseudopodia like structure, then absorption, then assimilation. Absorption is the digestion of the food will get digest, and that complete material will get absorbed by amoeba. Assimilation is generation of energy, that assimilated food will get absorbed and complete generation of energy will takes place. And egestion is nothing but the undigested food is then sent out through the surface of the cell. So one more time I am going to repeat it. First is the ingestion, then digestion, then absorption, assimilation, and then egestion. So these are the 5 processes. Okay. So next is the nutrition in human beings. So we will see it in next presentation.